Our last speaker in this session is Dr. Debbie Cory-Slechta. She is currently a professor at the University of Rochester. The list is just too long of all the amazing leadership positions that she has had. She is a former dean for research and chair of the Department of Environmental Medicine. She was formerly the lead of the Environmental and Occupational Sciences Institute in New Jersey, where she was also a department chair. She has been the lead on not one but two NIEHS Centers of Excellence in Research. She currently directs the K-12 WHEEL program in women's health and the environment over the entire lifespan. She is in much demand as an advisor to many of the NIH institutes, including the National Institute of Environmental Health Sciences, the National Center for Toxicologic Research, the FDA, and the EPA. She's going to tell us today about some of her really exciting research on links between air pollution and neurodevelopmental disorders. Debbie, thank you so much.
Well, thank you, Cheryl, and thank you to the organizers of this meeting. I'm going to start by, as soon as I figure out how this works. Nope. That's that. Okay, I'm going to start by giving you a quick crash course in air pollution because I'm concerned that many of you don't know about it. Obviously, the primary source is traffic generated. We have primary versus secondary sources of air pollution. At its base, however, what you have to remember is particles that have a carbon core to which there are organic and inorganic adherents to these particles, to this elemental carbon core. It is driven, the toxicity of air pollution is actually driven by the size of particles, as is the regulation of air pollution. So we talk about particle sizes in three different ranges. One is less than 10 microns. These are coarse particles regulated by EPA. You can see here a diagram. Oops, that's what I didn't want to do. Okay. There we go. A human hair, and you can see the fine particles. This is now less than 2.5 micron relative to a human hair. This is fine particles. And then finally, we get to what are called the ultrafine particles, and these particle sizes are 100 nanometers or less. And the smaller the particle, the more toxic it is, because this ultrafine particle has far greater surface area for adherence of different toxic contaminants. Toxic contaminants, if we think about them during development, can reach the fetal brain in many, many different ways. These particles, particularly the ultrafine, which seem to be able to bypass macrophages and other mechanisms for breaking them down, can in some cases cross the placenta, as can the adherents on those particles, which can also be dissolved and cross the placenta. From there, they can get into the fetal bloodstream. At this point, we're at a point where a lot of the regulatory mechanisms in the brain are not well developed, so in a way, there's an open pathway into the fetal brain. After birth, there are also several different mechanisms of exposure that can occur. Clearly, breathing in is one of these, and from there, the particles get into the bloodstream and can move to other organ systems through the circulation. But two other important routes that we need to think about, particularly in terms of lifelong exposure, is the fact that these particles can also be taken up via the olfactory nerve, including the contaminants that are on them. From there, they get directly into the brain, so they're bypassing the blood-brain barrier and not necessarily being then reflected in a peripheral measure of what's going on in your brain. In addition, we believe that particles can travel from the vagal nerve into the brain stem, so lots of mechanisms by which these particles can reach the brain. So I'm going to talk about these in relation to the brain. We know air pollution is considered the 6th leading cause of mortality in the world right now by the World Health Organization. That is based strictly on its cardiopulmonary toxicity. It really only has been in the last 5 or 10 years that there has been any attention being paid to what's going on in the brain, as if somehow it wasn't connected to the rest of the body and the bloodstream didn't get there. And so if you look at things, when some of the first epidemiological work that started coming out tied different metrics of air pollution to the diagnosis of autism spectrum disorder. And I think, in fact, now there's something like 15 epidemiological studies that have found those kinds of associations. And what they don't see, what the phenotype of autism spectrum disorder is, for those of you who aren't familiar with it, it's actually diagnosed on the basis of its behavioral characteristics, which include social and communication interaction deficits. You see repetitive behavior, impulsive kind of behavior. It can be accompanied by cognitive deficits and other comorbidities. There are, of course, multiple mechanisms that we think of in terms of autism, redox metabolism, excess glutamate, the glutamate theory of autism, inflammation, mitochondrial dysfunction, excess testosterone, this is a male-biased disease, and astrocyte dysfunction, and more recently, people have talked about the gut microbiome. So I'm going to show you changes from postnatal exposures of mice to ultrafine particle air pollution, and look at them in relation to some of these mechanisms. And what you're going to see in mice are very consistent with many of the features that we see in autism. First is ventriculomegaly. This is enlarged lateral ventricles, aberrant development of white matter tracts, and essentially dissolution of some of those, leading to interhemispheric disconnectivity. You'll see very persistent, long-lasting microglial activation, lifelong elevations of glutamate in these animals, and behavioral features that look very much like autism, and a male-biased pathology here. So we do this actually because we have an inhalation core facility where we can pull ambient air in from outside and separate it by particle size and deliver it to animals in these exposure chambers. We do this, what I'm going to show you right now, from postnatal days 4 through 7 and 10 to 13. We do these exposures for 4 hours a day. So to the ultrafine component, 4 hours a day, early in the morning when most of the traffic is there. The reason we chose those days is because this is considered equivalent to human third brain trimester development. It's a period of very rapid neuroangiogenesis, as you can see in the bottom here. So this was what really got me interested in air pollution when I saw these sections that came back from after we had done some of these exposures. On the top, what you can see are coronal sections from the brain of an air exposure, a controlled filtered air exposure animal, and you can see I showed a lateral ventricle here. These are brains from, from a male, from a male that's been exposed during this time period to ultrafine particles. And I think the ventriculomegaly is quite clear. What's going to become important here too, on top of this, of course, is the corpus callosum, the largest white matter tract of the brain, which in some other, in fact right here you can see, it's very sparsely cellularized there. This was one of our first studies. We saw these effects out until at least postnatal day 270. Those were the last brains that we had from that. Because we're at the mercy of whatever the ambient air pollution level is outside, we can concentrate it, but we're still at a base of what's outside. We've now seen this ventriculomegaly, which is male-specific, even at an average of 44 micrograms per cubic meter, so a very relatively low level of exposure. There are also aberrant white matter development that goes on in that corpus callosum, which is here and here. It's the largest white matter tract in the brain. It connects the two hemispheres and is critical for interhemispheric connectivity. And here again, you can see in the males, this is at postnatal day 14, right after these exposures end. You can see the significant reduction in myelin in the corpus callosum, and you can see it in these figures up here as well, that we didn't see in the females. Now, this effect did tend to go away by postnatal day 270 in these animals. We also saw reductions in the size of the corpus callosum, again, specifically in males, as well as microglial inflammation. So here's the reduction in the size that we see at postnatal day 14 in that corpus callosum, along with the white matter, loss of white matter. And even at postnatal day 270, that's when this data was taken from, there is a 340% increase in microglial inflammation still in the corpus callosum. So clearly these effects are not just sex-specific, but they're very persistent. We also saw an increase, a lifelong increase in glutamate in these animals. You can see, again, male-specific effects, slight effects in females, but they don't really become significant as opposed to males, where this difference seems to actually increase over time, at least in the time points that we have so far. We've also looked at behavior. One of the things we wanted to look at was impulsive kind of behavior, ability to inhibit responding. This is a task that we developed initially for rats and then use it now for mice. We allow them to get free rewards, but in order for them to get free rewards, they first have to start by making a total of 50 responses on a lever in an operant chamber. And that actually starts a period of free reward delivery. So you don't have to do anything, and you keep getting food pellets. The trick here is that the time between these free food pellets just keeps getting longer and longer. And so if you want to shorten that time period, you have to make another 50 responses to go back to these short time periods. And I think this slide demonstrates pretty clearly what the difference is between filtered air and postnatal ultrafine particle exposed males in this particular case. You can see this animal made its 50 responses and it just keeps collecting food deliveries, eventually starts over again. And here you can see with ultrafine particle exposure, there's virtually no ability to wait. They keep resetting and resetting that 50 response requirement. So the number of responses that they're making for each food delivery is extraordinarily high. We've also looked at social behavior. In this case, we were looking at the preference of an animal for either a novel conspecific or its cage mate. And what we find in those cases is that the males selectively show a reduced preference for a novel conspecific, somebody they don't know, an effect that we don't see with their cage mate. We also have found, we haven't really pursued this yet, that these air pollution exposures change levels of testosterone in males. So what accounts for male vulnerability? This is a whole story by itself. One of the things we believe is that there are activated microglia colonizing male brain. This occurs earlier in males and females. These microglia need to be activated for gliogenesis and neurogenesis, but of course it also makes them particularly vulnerable to an inflammatory stimulus. I'll show you a few slides about gestational exposure. So this is essentially trimesters 1 and 2. Here again, you're going to see the ventriculomegaly. In this case, we see it in both sexes, not just in the males. In this case, however, the effect was persistent in females, as you can see down here at the bottom, but not in the males. The other thing that we saw was a hypermyelination. So remember, when you do postnatal exposure, there's too little myelin and a small corpus callosum. Here, there was an increase in the size of the corpus callosum and way too much myelin happening way too early in these particular brains. Increases in activated microglia, again, in both sexes, as you can see in the top slides for the females and the males. There's an increase in the number of these activated microglia, again, a risk factor for an inflammatory stimulus. We know that these gestational exposures also produce damage to the myelin sheath, and I think you can see where these yellow arrows are, where some of that damage is occurring. We've also started to ask questions about what is contributing to this. So we've done a couple of different exposures where we only expose the animals to that elemental carbon core. So we strip off everything and just do the carbon core. That doesn't produce the neurotoxicity. And we've also generated aerosols of endotoxin, together or alone, and that also does not reproduce this. So I want to just show you in a second or two where we're going with this now. These are XRF analyses from filters from these exposure chambers for inorganic trace elements, and I think you can see very high levels of sulfur in these particular exposures, as well as iron. And I think if I skip forward, these form agglomerations in the brain, whole nother story. These are sagittal sections that have been subjected to ICP-MS, and I think on the top what you can see is the very high levels of iron in these brains. These are from a male, as well as the very high levels of sulfur that are occurring in response to these exposures. And so that's our next direction. We're going to be generating aerosols of iron dioxide as well as sulfur dioxide alone and then putting them together. But the message I want to leave you with is I've talked about this largely in respect to autism, but it's very important to remember that while every neurodevelopmental disorder has some unique feature or features, they share many, many different features. And this is just a list of some of those. The male dominance is seen very frequently in neurodevelopmental disorders, as are things like white matter changes and inflammation, altered glutamate function. So I think we have to think about these risks as not just being autism, but much broader. And I want to close by thanking all the folks in my lab who've been working on this, and in particular Günter Oberdorster, who is Mr. Ultrafine Particle, for those of you who are familiar with this area of research, and really dragged me into this, and it's been a great ride. So thanks very much, and again, thanks to the organizers.
For a couple of questions, but I'm just curious, Debbie, is it possible or has it been looked at in the brains of autism patients whether or not there are any of these metals or signs of exposure?
Yeah, so she asked me about whether people had looked at, that's a really interesting question for a number of reasons, looked at trace metal levels in brains of autistic individuals. Basically, more what's happening is taking blood levels, and if you look at the literature, it actually suggests that iron levels are lower in autism. However, if you then look at diseases, like there are genetic diseases that cause brain iron overload, where you'll have elevated brain iron, but lower plasma iron and ferritin, et cetera. So clearly there's a disconnect between what's happening in the brain and what isn't. The other thing I think that's so important about iron is, especially up the olfactory nerve, is that it has a half-life estimated to be on the order of decades. So once it's up there, it isn't going anywhere. And you need to think about that, or I've started to think about that a lot in terms of things like Parkinson's and Alzheimer's, where there's this clear accumulation of iron for reasons that aren't well known, have been suggested to be mechanistically involved with mechanisms like ferroapoptosis. And so I think you're going to start to see a lot more work on that as well.
And what about as a driver of inflammation?
Oh, the metals? Well, that's another one. Between the iron and the sulfur, we're now into the redox cycle, which is very active in the brain. And so if we already know, I didn't show you pictures, that we have changes in glutathione in those brain levels as well. So it's another potential mechanism. Yeah.
All right.
Thank
Well,
you.
Wonderful. Thank you so much. And let's have another round of applause for all those people.